Welcome everyone. Welcome to another video of BioGuide. Hope you people are all well. And today we are going to discuss about something new topic and which is very proud moment for all of us because Nobel Prize 2020 in chemistry has been awarded on this topic, which is CRISPR-Cas, new era of molecular editing. So what is CRISPR? Full form of CRISPR is clustered, regularly interspaced, short palindromic repeats, and it is accompanied by an enzyme which is called Cas9. It's a technology which is a revolutionary gene editing method in which a programmable RNA guides a nuclease to find a specific target location in the genome. How it was discovered and who has been discovered. We all know everything about this, who discovered this. This woman, Jennifer Doudna, in 2013, along with her colleagues, developed this CRISPR-Cas gene expression system, and when it was introduced into animal cells, it made site-specific changes into intact genomes. And it is very much precise, efficient, and less expensive than any other genome editing tool. And as a result, it has facilitated a wide range of studies that were previously unachievable. So if you go to the evolutionary history or the existence of the system, it was already there in the nature within bacteria and archaea. They use this CRISPR-Cas system for the adaptive immunity. This CRISPR locus, it consists of short palindromic repeats, and these palindromic repeats are separated by short non-repetitive sequences. So basically, it is a sequence of DNA in which there are palindromic repeats. We all know what are palindromes means. If you read these sequences from the left to right as well as from the right to left, both seems the same. So these palindromic repeats are interrupted by short non-repetitive sequences which are called the spacers. Spacers originate from invading sources of DNA such as phages that are copied and incorporated into the CRISPR locus when an infection occurs. This means suppose in bacteria, a phage has infected it and inserted the phage DNA into the body of bacteria. Now the bacteria within its CRISPR locus, it will the phage DNA will be incorporated into the repeats which are present, and thus this creates some spacer sequences and they are generally incorporated into them. Next, it contains two components. Basically, it contains a Cas, which is a molecular Caesar that can cut the double-stranded DNA at specific location. And obviously, it contains a guide RNA. This guide RNA contains two parts, a longer RNA scaffold along with a shorter stretch of RNA with pre-designed sequence about 20 nucleotides long. Together, they form CRISPR-Cas complex. So here, if you see the structure in the bacterial circular chromosome, the black portion that has been shown here in enlarged view, this contains some of the sequences which starts with the leader sequence. Then these are the CRISPR repeats and which are interspersed by the CRISPR spacers. And here they contain in the leader region some Cas genes. So this is the basic structure of CRISPR-Cas locus within bacterial genome. While how does this work? At first, the Cas9 has to locate. The Cas is the particular enzyme that will locate the particular sequence where the cutting or the editing has to be done. So it bind to a particular common sequence within a genome, which is called PAM sequence. Once the PAM is bound, the guide RNA unwounds the double helical DNA. And the RNA strand is designed to match and bind with a particular sequence within the DNA. Now, the Cas breaks the DNA with its nuclease domain, which can cause double-stranded break, and the DNA repair machinery would try to repair this double-stranded break, but this repair is error-prone using NHEJ or non-homologous end joining mechanism. As a result, which can be used for precise gene correction or insertion. Suppose you have a particular double-stranded DNA and you want to insert your gene of interest within that particular DNA. For this, what you can do, you can use the CRISPR-Cas9 system. Your Cas9 will locate the particular common sequence within the DNA, which is called PAM. Now, the guide RNA will cause the entire DNA molecule to be unwounded, and then another RNA, which is complementary to your target sequence, there it will bind, and by making it certain that this is the region it has to be cleaved. Now, your Cas enzyme, which is a nuclease enzyme, it will cause the double-stranded break and create a gap between it. In this gap, this is your place to insert new gene of interest. Here is the structural view. See, suppose this is your gene, your DNA or DNA of the animal in which you're working. Now, in this particular, this is your target sequence. Now, you can see this is the guide RNA and also the tracer RNA. Initially, the tracer RNA binds with the CAM sequence, and then the guide RNA guides and have complementary with the target sequence, which make it a certain that yes, this is the sequence where the cleaving has to be done. Now, this Cas, which actually does two purpose, one to scaffold the region as well as to cut the ends of the two double-stranded DNA, and thus after cutting it will create the gap, which is the place of inserting your gene of interest. It's a great tool for any knockout study, or in the case of other genetically modified organism, you can use it for activation of transcription. Suppose a repressor sequence is present, which is hindering a particular gene to transcribe. You can remove that repressor sequence using this CRISPR-Cas9 system. So then even in the gene silencing, suppose there is an enhancer sequence which allows a certain gene to express, but if you remove that enhancer aside using CRISPR-Cas9, your gene will be silenced. So these are very, very important. Even if you can use fluorescent molecules, you can even trace your gene of interest, its locus within the body using the CRISPR-Cas9 system. So there are several possibilities in which alteration of genome sequences can occur with the help of CRISPR-Cas9 system. Researchers are trying to utilize the versatility of this complex in therapeutic and environmental purpose. So thanks to this, to famous personalities, although I know all of you now know all of them. The left one is Emmanuel and the right one is Jennifer. Thanks to this woman for giving this gift to the research field, and thanks to all of you for listening to my video till the end, and hope you have a healthy, happy life and keep on having an essence of science and grow a scientific mentality. Thank you very much.